Hello guys, in this chapter I want to talk about the nervous system. Throughout the body, your body perceives and responds to multiple sensations. You smell spring flowers, feel the touch of a hand on your shoulder, and are visually aware of environment. You control multiple muscle movements to walk, talk to the person sitting next to you, and hold this textbook. Other muscle movements occur without your voluntary input. Your heart beats, your stomach churns to digest your breakfast, and you jump at the sound of a warning horn. All of these sensations and muscle movements are interpreted or controlled by the nervous system. In this chapter, I want to introduce the nervous system by first describing its general function and overall organization, and then discussing the component of nervous system, which is the primary tissue of nervous system. And the next several chapters investigate different aspects of nervous system, including the structure and function of the brain and cranial nerve, spinal cord and spinal nerve, and autonomic nervous system. Now, about this chapter, I want to describe three general functions of nervous system, identify the structural component including the CNS and those which are located in our PNS, explain the functional organization of nervous system, describe the structure of a nerve, including the three layers of connective tissue wrapping, and explain how nerves are classified structurally and functionally. Before I start talking about the nervous system, I want to talk about the systems which can control our body. We have two systems which can control the different functions of our bodies. One of them is the endocrine and the other one is nervous system. The endocrine system can do its control by different endocrine hormones, and the nervous system can do its function by secretion of different neurotransmitters. We have some differences between endocrine and nervous system. One of them is their means of communication. Look at this picture. In this picture, you see that in the nervous system, we have a release of neurotransmitters. Neurotransmitters are released from our neurons and they can work on the target cells. But in the endocrine system, we have a release of hormones. And you see that here the hormones can go to our bloodstream. By the blood, they can move in different parts of our body and then they can act on different cells. So you see that their means of communication are different. And you need to know that in the nervous system, besides releasing of neurotransmitter, we have some electrical methods which can communicate between different cells. The other differences between nervous system and endocrine system is the speed and persistence of the response. The nervous system can react quickly and stop quickly. Imagine that you put your finger on a flame of a candle. Immediately, you take your finger away and everything is finished. But the endocrine system reacts slowly because we need to release of hormone in seconds to days, and the effect may continue for weeks. Imagine that your blood sugar come down. Some receptors in your body should detect it, send information to your pancreas, and your pancreas should release a hormone which we call it glucagon, and glucagon should increase the amount of sugar in your blood. All of this process takes time. And when your blood sugar go to its normal level, then it continue to be in this level for several weeks. The other differences which we have between nervous system and endocrine system is adaptation to long-term stimulation. Your nervous system can adapt quickly and immediately the response declines. But the response of endocrine system has more persistent response. And the area of defect are different. The nervous system affects on a specific organ or a specific cell. But the effect of our endocrine system is general, widespread in all parts of our body because it can move by blood to different organs and it can only act on the organ or on the cell which has its own receptor on it. So you see that we have differences between endocrine and nervous system, but both of them wants to control our body function. And now I want to talk about the nervous system in this chapter. What are the general function of this system? Here you can see the general function. Our nervous system can collect information. The receptors are a specialized nervous system structure that monitor changes in both the internal and external environment of our body, which we call these changes as stimuli. For example, receptors which you have in your skin can detect the stimuli associated with touch. This sensory information then is relayed along neuron to your spinal cord or brain. The other function of our nervous system is process and evaluate information. After processing sensory information, the brain and the spinal cord determine what response is required. And then we have initiation of response to different information. The brain and the spinal cord initiate a response as motor information is relayed along neurons to a structure which we call them effector. Effectors include all three types of muscle tissue and glands. The effect may be either muscle contraction or a change in the secretion of the gland, and I will talk about it later for you. So pay attention. All the times, the effector are three types of muscles. Our smooth muscle, skeletal muscle, and cardiac muscle, and different types of glands. Some of these glands are endocrine, and some of these glands are exocrine glands. So our nervous system should detect the stimulation, process the information, and then initiate the response. And you can see all of this function in this picture. You see that you can see the information of this cup of water by your eyes. Then this information should move by sensory input or sensory neuron to your brain or spinal cord. They are your integration center. They process the information, evaluate the information, and initiate the response. And the response is muscle contraction here to grab a cup of water. So we have some motor output. They go to our skeletal muscle. The skeletal muscle go to contraction, and you can grab this cup of water and you can start to drink. This is the function of our nervous system. For our nervous system, we have two types of divisions. The anatomists and physiologists have devised various ways to organize the structural and functional component of the nervous system. So I want to talk about these two different organizations, structural and functional organization. Look at here. Here you can see the structural organization of the nervous system. Structurally, we can divide our nervous system to central nervous system or CNS and peripheral nervous system or PNS. The central nervous system, as you see in this picture, include the brain and the spinal cord. The brain is enclosed in our skull, and the spinal cord is located in our vertebral column. And the peripheral nervous system includes nerves. All of these yellow lines which you can see in this picture are nerves. They are bundles of axons. And also we have some ganglia. Ganglia are the cluster of neuron cell bodies which are located along the nerves. Nerves and ganglia are described more later for you. So this is the structural organization of the nervous system. And now I want to talk about the functional organization of the nervous system. Functionally, we can divide our nervous system to sensory nervous system or afferent nervous system, which can receive sensory information from the receptors and transmit it to the central nervous system. And the other subdivision is called motor nervous system or efferent nervous system. They can initiate response to our effector. And now I want to talk about this functional organization. Now look at this picture. In this picture, you can see these two divisions of nervous system. One of them is sensory part and the other one is the motor part. I want to talk about sensory part. The sensory nervous system can detect a stimulation and transmit information from receptors to the central nervous system. We have two types of receptors. Some of our receptors are our five senses. You can see, you can hear, you can smell, taste, and sense. All of these five senses can be detected from external environment. So, if you have any stimuli, if you have any changes in your external environment, you can detect them by these five senses. The information of these five senses are called somatic sensory information. Somatic sensory information are the information which you can get from outside. Sometimes you have some information inside of your body. For example, your blood pressure increase or your blood pressure decrease, or you have some changes in the content of your esophagus, in the content of your stomach. So you can receive these different stimulation from inside of your body. And you cannot control the function of structures which you have inside. So the information which you can get from inside are called visceral sensory information. Up to here, I talked about sensory nervous system, which can detect a stimulation. If you detect a stimulation from outside, we call it somatic sensory. And if we don't want to hear, we can close our ear. If we don't want to see, we can close our eyes. And the other information come from inside of our body. We detect information and receive information from inside. We call it visceral sensory. Somatic sensory from outside and visceral sensory from inside. Now, all the information from outside and inside move to our central nervous system, which is the brain or the spinal cord. The integration and information process take place in our central nervous system, and then this information should move to our effector organ by some errant neurons or motor neurons. which we call it motor nervous system. The motor nervous system can initiate and transmit information from the CNS to our effector organ. If this information go to our skeletal muscle, pay attention here, if they go to our skeletal muscle, we call it somatic motor. If this information go to smooth muscles, cardiac muscles, and glands, we call it autonomic motor. What is the difference between somatic and autonomic? Somatic motor is voluntary. If we don't want to move our muscle to contract our skeletal muscle, we can do it. But for the autonomic motor, it's completely involuntary. You cannot control the function of your heart. You cannot control the function of your intestine or stomach. And you cannot control the function of your thyroid gland, your pancreas, or your parathyroid gland. So we call it autonomic motor. So the motor nervous system, which can initiate and transmit information from central nervous system to effector, is divided to somatic and autonomic. Somatic can go to a skeletal muscle only and do contraction and relaxation, and autonomic motor go to cardiac muscle, a smooth muscle, and gland. It's completely involuntary. And I will talk about autonomic motor in chapter 15 completely. The autonomic motor is divided to two subdivisions. One of them is called sympathetic or fight or flight, and the other one is called parasympathetic or rest and digest, which I will talk about it later for you. So I talked about the structural organization, which is the central nervous system and peripheral nervous system, and then I talked about the functional organization, which we have sensory nervous system and motor nervous system. The sensory nervous system can receive information from outside, which we call it somatic sensory, by our five senses, and from inside, which we call it visceral sensory. All the information go to our brain and the spinal cord, and after that our motor nervous system start to initiate and transmit information to the effector organ. If the effector organ is the skeletal muscle, we call it somatic motor. If the effector organ is the cardiac muscle, a smooth muscle, or glands, we call it autonomic motor. Now, I want to talk about the nerve structure and the nerve anatomy. A nerve is an organ composed of a cable-like bundles of axon, connective tissue layer, and blood vessels. And the nerve is a component of our peripheral nervous system. Here you can see the organization of the nerve. First, I should tell you that the axon is one part of the nerve cell or neuron, which I will talk about it later for you. Look at here. In this picture, you can see the axons. Each axon is covered by the areolar connective tissue. This areolar connective tissue is called endoneurium. So, endoneurium is the areolar connective tissue which can wrap the individual axon and separate axons from each other. A group of axons are covered with the other connective tissue, which we call it perineurium. This perineurium is a dense irregular connective tissue, and the perineurium can make a nerve bundle or nerve fascicle. The fascicle is bundles of axons which we have in the nerve. And a group of fascicles are covered with the other connective tissue, which is dense irregular connective tissue, and it's very thick, and we call it epineurium. So, individual axon is covered by endoneurium. A group of axons are covered by perineurium and make nerve bundle or nerve fascicle, and a group of fascicles are covered by epineurium. But why do we need these connective tissue around the axons and around the nerves? Because the connective tissue contain many blood vessels, and our nerves are vascularized by an extensive network of blood vessels which branch and extend through both the epineurium and the perineurium to become capillaries. Capillaries are associated with the endoneurium and function as the site of exchange of oxygen and nutrient for waste product and carbon dioxide between the axons and the neurons and the blood vessels. So here you can see the nerve anatomy. We have the other picture here. In this picture, again, you can see individual axons which are covered by endoneurium. Then a group of axons are covered by perineurium, and then they can make fascicles or bundles, and a group of bundles are covered by epineurium. When we come from endoneurium to perineurium and then epineurium, the connective tissue becomes denser and thicker. And you can see the blood vessels which we have in these connective tissue. And here you can see the scanning electromicrograph of the nerve. You can see the axon which are wrapped by endoneurium and a group of axons which are wrapped by perineurium. And between these fascicles, you can see many blood vessels. But in the other picture, in the other part, you can see the ganglia. And what is the ganglia? A ganglia is a cluster of neuron cell bodies. These round structures are cell body of neurons, and all of them are gathered together out of our central nervous system. Pay attention. The ganglia is the cluster of nerve cell body in our peripheral nervous system, and they are wrapped by connective tissue. The cluster of cell body result in a swelling or enlarged portion, look at here, enlarged portion along the length of nerve, which is often large enough to be observed with the naked eye. A specific type of ganglia include the dorsal root ganglia associated with sensory neuron that extend into the spinal cord and are associated with the spinal nerves, and the ganglia associated with motor neuron that extend to autonomic effector for the parasympathetic and sympathetic division, which I will talk about them later. For now, you need to know that the ganglia is the cluster of nerve cell body in our PNS. You can see them as a bulged portion or swelling along the side of our nerves. The nerves are classified to two parts. One of these classification is cranial nerves. The cranial nerves are the nerves which extend from our brain. And we have 12 pairs of cranial nerves. And the other part of this classification is the spinal nerve. The spinal nerve extends from our spinal cord. and we have 31 pairs of nerve from our spinal cord. So we have 12 pairs from our brain, and we have 31 pairs from our spinal cord. And our nerves can be classified functionally. We have sensory nerves, which only contain sensory neuron, which can send information and signal to our central nervous system. We have motor nerves, which contain motor neuron, which can send signals from our CNS to effector organ. And some of our nerves are mixed nerves. It means that they contain both sensory and motor neuron. And most named nerves, the cranial and the spinal, are in this category. They are mixed nerves. So structurally, we can divide our nerves to cranial and spinal, and functionally, we can divide them to sensory, motor, and mixed nerve. And as I told you before, the gang is a cluster of neuron in our peripheral nervous system. Pay attention, it's located in peripheral nervous system. And now you can answer this question. What is the function of receptor? And what are the different types of effector controlled by the nervous system? What are the two primary functional divisions of the nervous system, and how do they differ from each other? And what are the three connective tissue wrapping in a nerve, and what a specific structure does each of them initiate? Now, I want to talk about the neurons. The nervous tissue is the primary tissue of the nervous system and is composed of two distinct types of cells. One type of cell is called neuron, the functional cells which we have in the nervous system, and the other type of cell is called glial cells or neuroglial cell. The glial cells are non-excitable cells which can do protection, nutrition, and suppression of our neurons. Now, I want to talk about neuron, the functional unit of our nervous system, the basic structural unit. This neuron has a different characteristic as you see here. The first characteristic is excitability. Excitability is responsiveness to a stimulus. This stimulus can be chemical, stretch, or pressure changes. The stimulus caused a local electrical change in the resting membrane potential in the excitable cell by initiating the movement of ion across the plasma membrane, which I will talk about it later for you. So the neurons are the cells which become excitable. They can react electrically to different stimulation. The other characteristic of neuron is conductivity. Conductivity is ability to propagate the electrical signals. When our neuron becomes stimulated, this stimulation can move and propagate it in the length of our neuron, in the plasma membrane. And from the beginning of the neuron, it can move to the end of neuron, and then it can go to the other neuron. We call it conductivity. The other characteristic is secretion. Secretion is the release of neurotransmitter in response to conductive activity. When the information from our neuron reach the end of the neuron, we have a release of neurotransmitter, and this neurotransmitter can affect the next cell or the next neuron. The other characteristic of neuron is extreme longevity. It means that most neurons form during fetal development are still functional in very old people. And the neurons are amitotic. It means that they cannot do mitotic division. And for example, for any reason, if we lose some of our neuron parts of our nervous system, that part cannot regenerate the neuron. Only that part is filled with a connective tissue and glial cells. These are the characteristics of neuron. So the neuron can become excitable. They can conduct the electricity and the electrical signals, and after conductivity, they can do secretion, and they can live for the lifetime, and they are amitotic, and they cannot do any mitotic division. Here you can see the structure of the neuron. The neuron has different parts. The first part and the important part of our neuron is the cell body of the neuron. And here you can see the cell body. In the cell body, we have nucleus. In the nucleus, we have nucleoli. And you can see many organelles in the cell. Just we don't have one organelle. And that organelle is the centriole, because you know that the centriole is the organelle which can start mitotic division, and we don't have mitotic division here. We have some extension which come to our cell body. These extensions are called dendrites. The dendrites tend to be relatively short, a small tapering process that branch off the cell body. Some neurons have many dendrites, some of them has only one. Dendrites, like the cell body, are not insulated with the myelin sheath, which I will talk about it later for you. And then we have a long extension from the cell body, which we call it axon. And we have only one axon per each neuron. The single axon is typically a longer process from the cell body to make contact with other neuron, muscle cell, or gland cells. A specialized terminology is used for both the axon cytoplasm and its plasma membrane. The cytoplasm, which we have inside the axon, is called axoplasm, and the plasma membrane of the axon is called axolemma. The axon may be is insulated by a myelin sheath, and this whitish part which you can see around the axon is the myelin sheath. The myelin sheath can insulate our axon, and it doesn't let the information move to the neighboring axons, and I will talk about it later for you. At the end of the axon, you can see telodendria or axon branches, and then we have axon knob, which is a swelling at the end of the axon, and I will show you them better in the next pictures. Again, in this picture, you can see the cell body of our neuron. Here are the dendrites which branch off from the cell body. They are tapered, as you see here at their end, and they are short process. Here you can see a single extension from the cell body, and we call it axon. And at the end of axon, we have axon terminal. Now I want to talk about the cell body. The plasma membrane encloses the cytoplasm of the cell body. The other name of cell body is perikaryon. It contains, as I told you before, a nucleus, and our nucleus contains nucleoli. We have some structures. Let me show them to you in the next picture. I don't know if, yes, here. We have some structures in our cell body which we call them Neisel bodies or Neisel substances. The Neisel bodies are the RER and the ribosomes in our neurons, and they are chromatophilic structures. Chromatophilic structures means they like to attract a stain, and when we stain and color our neuron, they can get the stain and they can show themselves. And these structures are very important for our neurons because, as you know, the ribosomes and the RER are the organelles which can make protein, and the protein which is made in our neuron are neurotransmitters, and neurotransmitters are important for communication between one neuron and the next neuron. And as you see in this picture, the neuron contains all other structures. You can see Golgi apparatus, you can see polyribosomes, you can see microtubules, you have mitochondria, a smooth endoplasmic reticulum. So we have all the organelles in our cell body or perikaryon. In this picture, you can see the dendrites. The dendrites are short, unmyelinated, tapered process which branch off from the cell body, and then you can see the axon. When our axon wants to begin, we have a triangle portion. Let me show you this triangle here. This triangle portion, which is the start of our axon, and we call it axon hillock. So the axon is a start from our axon hillock, and it's a single process which come out of our cell body and continue to the next cell. The endoaxon branch, and we call them telodendria, and the tip of each telodendria becomes swollen, and we call it synaptic knob or terminal bottom. Synaptic knob or terminal bottoms. In the synaptic knob, we have many, here you can see the synaptic knob, and you can see many synaptic vesicles, and these vesicles contain neurotransmitters which are released from our axon. As I told you before, our axon contains the cell membrane which we call it axolemma, and inside the axon we have cytoplasm which we call it axoplasm. In the axon you can see microfilament and microtubules, and they are important for the shape of the axon and also a movement of a transmitter from the cell body to the end of the axon and vice versa. The function of the axon is conduct action potential and release neurotransmitter at the synaptic knob. So these are the different parts which we have in our cell bodies. And you need to know that in our neuron, we have cytoskeleton. The cytoskeleton composed of microfilament, intermediate filament, and microtubules. The intermediate filament are called neurofilament, and these neurofilament and all of these cytoskeleton can make this a specific shape which you can see for neurons. Again, in this picture, you can see the structure of neuron. You have a cell body or perikaryon. The other name, we have the other name as well. We call it soma. You can see many dendrites here. Then you have axon hillock, these triangle shaped structure, and then you can see the axon. And this axon is covered by myelin sheets. It's insulated by myelin sheet. And at the end of the axon, you can see axon terminal, telodendria or axon branches, and at the end of axon branches you can see synaptic knob or axon knob, which contain many vesicles. And the vesicles contain neurotransmitters. Here you can see the communication between this is the axon and the axon terminal and our neuron. When the information comes down in this axon to axon terminal, we have release of neurotransmitter from the synaptic knob to the next neuron. And this information are detected by the dendrites and the cell body, and then it can move to the axon and transmit it to the next neuron. And in this picture, you can see the histology of our neuron. This is the cell body. You can see the nucleus and nucleoli. You have these dendrites and axon hillock and then the axon. In this picture, you can compare the size of neuron, the functional unit of our nervous system, and these small cells. These small cells are called glial cells. And I told you that the glial cells can do protection, nutrition, and suppression of our neuron. In the neuron, and also, in the axon, we have a movement of a neurotransmitter. The neurotransport is divided into two categories. One of them is called anterograde transport. The anterograde transport starts from the cell body toward the end of the axon. And this is movement of neurotransmitter. And we have the other type of transport, which we call it retrograde transport. The retrograde transport is the transport in opposite direction. It starts from the axon terminal toward the cell body. And most of the times, the material which move in retrograde transport is for breaking down and recycling. And also, we have two types of transport in our axon. One of them is called a fast axonal transport, and the other one is called slow axonal transport. The fast axonal transport occurs about 400 millimeter per day. It involves movement along the microtubules, which I talked about it before for you, and powered by motor proteins that splits ATP. And this fast axonal transport can be anterograde or retrograde. It means that we have movement from the cell body to the end of the axon, or from the end of the axon to our cell body. And we have a slow axonal transport. The slow axonal transport occur about 0.1 to 3 millimeter per each day, and result from flow of axoplasm. Here our microtubules are not working, and substances only move from the cell body toward the knob. So we only have anterograde transport. The enzyme cytoskeleton component and new axoplasm can move by this type of transport. And in axonal transport, we have two proteins which are very important for this movement. One of these proteins is kinesin, which can drive the protein synaptic vesicles and mitochondria in anterograde transport. And we have a protein which we call it dynein, and it can help the neuron in a retrograde transport. We have different types of neuron. We can classify our neuron based on their structure and based on their function. Structurally, we can divide and classify our neurons to an axonic neuron, the neuron without any axon. We have bipolar neuron, which you can see two extension from the neuron. We have pseudo-unipolar neuron or unipolar neuron. If you pay attention to this picture, only one extension, which is very short, come of our cell body, and then it splits to two parts. And we have multipolar neuron, which is the common type of our neuron. And now I want to talk about these different classes. First, an axonic neuron. An axonic neuron doesn't have an axon, and the only thing which you can see are the dendrites. They are interneurons, which we have them in central nervous system, the neurons which are located between sensory and motor neuron, and actually the interneurons are the neurons which can do integration. The next type of neuron is multipolar neuron. Look at the picture. The multipolar neuron have many dendrites, as you see here, and they have a single axon that extend from the cell body. They are the most common type of neuron in the human body. All motor neuron and some of the interneurons are located in this category. The other type of neuron is bipolar neuron. The bipolar neuron has two extensions, one dendrite and one axon. The location of this neuron is very limited in human body. The neurons which we have in the retina of the eye and the olfactory epithelium in our nose and some parts of our ear are these bipolar neurons. They are sensory neurons. And the last type of neuron is called unipolar or pseudo-unipolar neuron. A single short process, as you see here, come off the cell body and then it divides into two branches like a T. These neurons are also called pseudo-unipolar or false unipolar because they start out as bipolar neuron during development. By their two process fuse into a single process. The naming of the branched process is in unipolar has been a source of confusion with regard to the common definition of dendrite and axon. It seems most appropriate to call the short, multiple branched receptive endings as the dendrite here, and the other portion is called axon, all the length. The axon is divided to two parts. One part is called peripheral axon and one part is called central axon. The peripheral axon is from dendrite to the cell body, and the central axon is from the cell body to the central nervous system. And most sensory neurons are located in this category. We call them unipolar or pseudo-unipolar. And the functional classification of our neurons are here. We have sensory neuron, which can conduct input from somatic and visceral receptor to the CNS. Most of them are unipolar and few of them are are bipolar, as I told you, in the ear, in the eye, and in our nose. We have a motor neuron which can conduct output from central nervous system to somatic and visceral effector, and all of our motor neurons are multipolar. And we have interneurons or association neurons. They can receive, process, and integrate information from many other neurons, and they communicate between sensory and motor neuron. They are located in the CNS, and they can make up about 99% of our neuron, and most of them are multipolar. In this picture, you can see these divisions. The sensory neuron can receive information from outside or inside of our body and move this information toward our central nervous system. Here you can see this neuron, and you know that this neuron is pseudo-unipolar or unipolar because we have only one branch which come out of our cell body. Then we have interneuron. The interneuron may be an axonic or multipolar neuron, and then you can see the motor neuron, which is the multipolar neuron, and start from the central nervous system and extend to effector organ. And here our effector organ is the skeletal muscle. So this is the functional classification of our neuron. Sensory bring the information to central nervous system, interneuron do integration, and motor neuron bring the information of central nervous system to the effector organ. And now you can answer these questions. You can explain the neuron characteristic of excitability, conductivity, and secretion. What are the function of these neuron structure? What is the function of dendrite, axon, synaptic vesicles, and neurofibrils? Each type of axonal transport is both anterograde and retrograde, and give examples of substances which are transported by this method. How are the different processes that extend from a cell body used to structurally classify our neurons, and where are interneurons located, and what is their function? In the next part, I will talk about the synapse and the glial cells.